figure out some stuff about DNA that moves from the mitochondrial genome to the plastid genome in this Monsonia, which is a species, a member of the geranium family. So what I want to do is I want to give you guys a brief primer on plant mitochondrial genomes, because I know most people in systematics and population genetics are really familiar with animal mitochondrial genomes, but plant mitochondrial genomes are very weird. And if you make the same assumptions when you're tackling evolutionary issues about plant mitochondrial genomes that you do with animal mitochondrial genomes, you'll probably be wrong. So they're highly variable in size. So unlike animal mitochondrial genomes, which, you know, the human mitochondrial genome is 16 kb, these guys vary in size from about 60 kb in the case of mistletoe to over 10 megabases in the case of species of Silene. And they're usually around 350 to 400,000 base pairs. They're also variable in gene content, though not as much. They have 20 to 40 protein coding genes. They have a low mutation rate, so they can't be used for low-level systematics like animal mitochondrial genes, like I mentioned earlier. They're about 100 times more slowly evolving. So this is about a third the rate of mutation rate in the plastid and a tenth the rate of those in nuclear genomes. And this is counterintuitive to what you know about mitochondrial genomes too. They're not recombinationless, they recombine a lot, and this facilitates lots of rearrangement and alternative configurations of the genome. And as an example of this is a plot of the genome arrangement in the 7 species that I'm looking at in Monsonia. Like, this is almost incomprehensible. It's like a Jackson Pollock painting or something. It's just completely chopped and screwed. So this 7-way comparison, almost uninterpretable. So let's just zoom in on 2 pairwise relationships. These 2 species, these 2 sister species pairs. Now these guys, it's interesting because these rearrangements are also variable. So both of these 2 splits are a little less than 1.5 million years old. And as you see, this guy is far less rearranged than these 2. So, yeah, so this top pair, this can be explained with about 20 rearrangements, and this bottom pair requires 95 rearrangements. So you have 95 block interchanges in under 1.5 million years. So, but this is remarkable, but what I'm going to be talking about most is during this talk is the incorporation of DNA from other compartments, the plastid, or other organisms, another angiosperm. I'm going to focus mostly on transfers from the plastid. So you all are aware, from animals and also plants, that DNA moves from the mitochondria to the nucleus, but you're probably less aware that it moves from the plastid to the mitochondria. So I'm going to be looking at this in this species, Monsonia. They're all small, nondescript herbs found in southern Africa. I've assembled the genomes of these 7 species. They're between 400 and 600 kb. In terms of gene content, and a lot of things are not variable, but one thing they are variable in is this plastid-derived content. Now, how this works is DNA, you have a copy of some DNA in the chloroplast, and it moves to the mitochondria. The mitochondria rearranges so much that these fragments get separated from each other and translocated to different parts of the genome. So the result is you have one copy in the plastid and one copy that's split somewhere in the mitochondrial genome. And this could happen like a bunch of times. So you could have one fragment that could be split into like 5 fragments or something like that. So I looked. So these are regions of homology that are shared by the mitochondrion and the plastid, mapped onto the plastid genome of one species, Monsonia speciosa. And as you see, there's a lot of these fragments that look like they're shared. So that's interesting that they come from the same spot, but how can we know that they're shared? That's one piece of evidence. The second piece of evidence is that if they share boundaries, and if you look at the boundary which 2 fragments in different species share, for example, like this one and this one, if you look at all of these fragments, if you look at 50 base pairs flanking those edges, 47 out of 50 are 47 out of 50 nucleotides that shares that much identity in the flanking region of each of these fragments. So you have 2 pieces of evidence that these guys were actually the result of the very same event. And we have a phylogeny of these species, so I can use a dowel parsimony to place these fragments on different branches of the tree. And then I can convert those fragments to the number of nucleotides. And we have a fossil deeper in this order so we can calibrate this and get a time tree. And so that's interesting. So we actually know when these fragments came in. Now there's a few interesting things on here. Among the most interesting is this guy right here, who seems to have had some invasion or series of invasion of plasmid, of plastid DNA in the last 1.5 million years or so. It doesn't seem to be correlated with that rearrangement we saw earlier, because if you look at all the pairwise comparisons, the vast amount of rearrangement that we saw in these 2 species down here is actually occurring mostly on the Monsonia ciliata end, rather than this end, which is experiencing this influx of plastid DNA. So we have the amount of plastid DNA that's come in on each branch, and we have a time tree. So what we can do is we can calculate a rate. Now this rate is the number of nucleotides which are plastid-derived per site per year in the mitochondrial genome. Now for a point of reference, if you look at the synonymous substitution rate in the Monsonia genomes, what you see is a rate that's about an order of magnitude lower. Now there's caveats when comparing these 2 numbers, but this should convey that this transfer of plastid DNA to the mitochondrion is a really, really reasonable, sizable force. And these other forces are forces we need to focus on, because a lot of time and energy is spent examining these single nucleotide events where you have processes like this plastid to mitochondrion transfer, which we have barely scratched the surface, but they seem to be having a comparable, if not greater, impact on the evolution of these mitochondrial genomes. So another interesting thing I discovered in Monsonia is you can have gene transfer from a different species, from one mitochondrion to the other. So this is a phylogeny of a particular gene, COX-1, of the angiosperms. I'm just going to go over this horizontal transfer briefly. But you have one copy which falls in the geranium family where you would expect it to, with high support, 100 support. And you have one copy of the gene, the mitochondrial genome, which is found all the way over here in the solanales. And it groups with this parasitic Cuscuta. It's a member of the morning glory family in multiple genes, 6 genes actually. So this intimate ecological interactions also facilitated the transfer of genes probably in this one species of Monsonia, but this is actually a whole other talk. But in summary, these Monsonia genomes have high and variable rates of rearrangement, and by using my approach, I've been able to infer hundreds of events revealing variable rates of plastic to mitochondria and DNA transfer. This is very significant because in the past, people have only been able to identify shared fragments into species a handful of times. So this is basically the first in-depth investigation of the evolutionary history of these fragments in mitochondrial genomes. And these transfers appear, if we take those values at face value, they appear to have introduced more nucleotides and single nucleotide substitutions. So this force is important. I'd like to stress that because this has not been examined before. And it's a very important aspect in the evolution of organelle genomes relevant to all flowering plants, all of which have some proportion of plastid-derived DNA in their mitochondria. And this horizontal gene transfer is actually only the 5th example in plants of multi-gene mitochondria and horizontal gene transfer in any plant ever. So there's lots of interesting stuff going on in these Monsonia mitochondrial genomes. And I'd like to thank these people. Does anyone have any questions? Yes?
Do the rearrangements in mitochondria ever cause,
The rearrangements in the mitochondria ever cause pseudogenes, or how are the borders of the fragments determined vis-a-vis the genes actually?
So the genes, you mean the mitochondrial genes? There's no overlap. There's no breakpoint, rearrangement breakpoint on a mitochondrial gene, but these plastid fragments are split by these rearrangement breakpoints.